The overall goal of this protocol is to differentiate human pluripotent stem cells into human trophoblastic cells using bone morphogenic 4 and inhibitors of the Activin nodal signaling pathways to study the early events of human placental formation and function.
This method can help answer key questions in the study of trophoblastic cell functions and how these cells are formed. The main advantage of this technique is that it can produce abundant trophoblastic cells for a wide variety of applications, including genetic gain and loss of function assays.
To begin the experiment, thaw an aliquot of extracellular matrix on ice for 3 to 4 hours. Using ice-cold tips, 1 cold 50 milliliter conical tube, and Delbecko's modified Eagle medium, or DMEM-F12 medium, transfer 2 milligrams of extracellular matrix into 24 milliliters of ice-cold DMEM-F12. Immediately transfer the 50 milliliter conical tube to ice and store it at 4 degrees Celsius. To coat tissue culture plates, add the 1 milliliter of diluted extracellular matrix per well of a 6-well plate. Swirl the plate to coat the wells and incubate the plate at room temperature for at least 1 hour. Aspirate the extracellular matrix from the new plate and add mouse embryonic fibroblast conditioned medium, or MEF-CM, containing BFGF. Use a pipette to transfer the scraped cellular clumps from the MEF plate and aliquot them into the extracellular matrix coated plate. Then, incubate the plate. Scrape off differentiated cells with a pasture pipette.